Hello, I'm Victoria Wong, and I'm a junior at Detroit Country Day School. I'm currently taking AP Biology, and I have always been fascinated by neuroscience and everything related to the brain. A few years ago, I was doing research about different neurological diseases when I stumbled upon diffuse intrinsic pontine glioma, an aggressive form of brain cancer. Ever since I learned about DIPG, I have been constantly checking on the research being conducted about it and waiting for updates about a cure. In this podcast, we will dive into the causes and symptoms of diffuse intrinsic pontine glioma and the potential cures that scientists have been developing over the past few years. So without further ado, let's begin our study of DIPG. At 6 years old, Erin was an energetic child. However, at 11 years old, she began to experience severe headaches, nausea, and vomiting. After getting a CT scan, she was diagnosed with DIPG, a rare brain tumor that develops in the brainstem and affects the nervous system. She was given radiation, chemotherapy, and surgery. Erin survived past her prognosis and began campaigning to raise awareness and increase funding for childhood cancer. However, in 2014, Erin deteriorated very quickly with headaches, poor balance, and seizures. She was hospitalized and lost the ability to speak, eventually passing away. Afterwards, Erin donated her tumor to researchers, hoping that the research would help develop a cure for DIPG. Diffuse intrinsic pontine glioma is a fast-growing malignant brain tumor that begins in the pons region of the brain. The pons is part of the brainstem and plays a vital role in controlling bodily functions, such as breathing, heart rate, blood pressure, and many other muscles throughout the body. Because the pons plays such a major role in controlling the body, the symptoms of DIPG become noticeable quickly, and the latency period is very short. DIPG is also different from other high-grade gliomas because it is very diffuse. Because DIPG spreads to the brainstem, cerebellum, and cerebrum, it is especially hard to treat and extremely deadly. Every year, around 300 kids are diagnosed with DIPG in the U.S. While DIPG can occur in younger children and teens, most of the diagnosed children are between the ages of 5 and 10 years old. Unfortunately, as of now, the survival rate for children with DIPG is very low. Only around 2% of the children live past 5 years after diagnosis, and long-term survival is very rare. Ultimately, the average time a child with DIPG survives after diagnosis is 8 to 11 months. Now, what exactly causes DIPG? DIPG is a glioma, and gliomas are caused by changes in the cell cycle of glial cells. Glial cells protect the brain's nerve cells and play a vital role in the brain, making this form of cancer particularly harmful. In general, changes to the cell cycle, such as the non-functionally regulating system, can cause the cells to divide uncontrollably. For example, mutated tumor suppressor genes, such as P53, a gene responsible for apoptosis, may cause the cell to be unable to undergo growth arrest and cell death. Oncogenes, which are mutated proto-oncogenes, can cause continuous cell growth and division. In the end, both the mutated tumor suppressor gene and the oncogene can cause cancer. Although scientists are aware of these mutations, the exact causes for the changes in the glial cells is unknown. While scientists have not quite determined the exact cause for DIPG, more research is currently being conducted. In a study by Michelle Monge and other scientists, DIPG has been linked to the dysregulation of postnatal development during middle childhood and is connected to an active hedgehog signaling pathway. The hedgehog signaling pathway is important in vertebrate embryonic development, and it controls the formation and morphogenesis of an organism. Monge's experiment studied cell populations with nestin, an intermediate filament protein, in the ventral brainstem and the pons. During infancy, cells with nestin are present in the ventral brainstem structure. These cells decrease by the age of 2. However, in the human pons, the density of these cells peaks around 6 years old, which corresponds to the incidence of DIPG. The hedgehog pathway is also part of the neural precursor cell populations. Scientists use beta-galactosidase to determine the presence of an active hedgehog pathway in the ventral pons. The beta-galactosidase positive cells were positive for OLIG2, a transcription factor known to be regulated in the embryonic spinal cord by the sonic hedgehog gene, and SOX2, a marker of primitive cell types. These cells increase dramatically in middle childhood. Thus, the hedgehog signaling pathway plays a role in the development of DIPG. To ensure that the hedgehog signaling pathway was causing the cancer, the scientists used the sonic hedgehog protein ligand, which activates the hedgehog signaling pathway. and they also used a hedgehog antagonist which represses the pathway. They then measured the red fluorescent protein produced from the hedgehog pathway. When the pathway was exposed to the hedgehog antagonist, the red fluorescent protein decreased by 67%. When the sonic hedgehog ligand was added, the red fluorescent protein increased by 200%, suggesting that the hedgehog pathway was indeed involved. Even though scientists found a correlation between DIPG and an active hedgehog pathway, the dysregulation of the hedgehog pathway doesn't necessarily cause DIPG. This is evident since some diseases have inactive hedgehog pathways, but the people do not develop DIPG. But a constantly active hedgehog signaling pathway is associated with the proliferation of DIPG cells. Now, the symptoms for DIPG are largely dependent on where the tumor forms, the size of the tumor, how fast the tumor grows, and the child's age. However, the symptoms develop quickly and include trouble with eye movements, vision problems, problems with talking, chewing, and swallowing, drooping on one side of the face, headaches, nausea and vomiting, weakness in the arms and legs, loss of balance, trouble walking, changes in behavior, and trouble learning in school. There are also many different methods for diagnosis. A magnetic resonance imaging, also known as an MRI, creates pictures of areas inside the brain. When gadolinium, a rare earth metal used for MRIs, is injected into a vein, it collects around cancer cells, making the cancer more prominent in the photo. Occasionally, a biopsy, specifically a stereotactic biopsy that uses imaging techniques to find and remove the tumor tissue, can be performed. However, because of the location of the tumor, it isn't always the best option. Finally, immunohistochemistry can also be done. It uses antibodies to check for antigens in tissues, and these antigens are often used to determine if the cancer is present. Currently, there are no cures for DIPG, but there are treatments that can be used to extend the patient's lives. Such treatments include radiation therapy, chemotherapy, and surgery. Radiation therapy uses high-energy X-rays and other forms of radiation to kill cancer cells. Since this causes changes in brain tissue, it can cause damage to the brain, affecting growth and brain development. Chemotherapy is another option that uses drugs to slow or stop the growth of cancer cells. It can either stop the cells from dividing or kill them altogether. Finally, surgery can be used to place a shunt. This is often needed for children with DIPG who have fluid around their brain or spinal cord. The shunt carries extra fluid away from the brain, which ultimately decreases the fluid and the pressure on the brain and spinal cord. Recently, scientists have also found several promising methods to combat the cancer. I'm going to talk about one specific method that L.M. Smith and other scientists have been studying: synchrotron microbeam radiation therapy. Recently, scientists have considered this microbeam radiation therapy as a potential alternative to the conventional radiation therapy being used to treat DIPG. Microbeam radiation therapy has been especially promising because it spares normal brain tissues by using spatial dose distribution. This reduces the damage caused to the brain by conventional radiation therapy, and it slows down the progression of the tumor still. Scientists have tested the microbeam radiotherapy for efficiency by examining apoptosis and the progression of the cell cycle in cancer cells that have been exposed to it. They found that in cell lines with an active p53, the gene that creates the protein responsible for apoptosis of the cancer cells increases significantly. Additionally, those cells were found to have more cells in the sub-G0 phase, which is also known as apoptosis, and decreased number of cells in the G0, G1, G2, and M phases of the cell cycle. Thus, apoptosis increased and the cell cycle decreased after the microbeam radiation. For cell lines without a functioning p53 gene, there was a decrease in the cells in the G0 and G1 phase. However, there was an increase in the sub-G0 and G2 and M phases. This accumulation of cells in the G2 and M phases suggests that there was a prolonged mitosis, so the DNA that was reduplicated was not separated correctly and accumulated in the wrong phase. Since the DNA was not separated correctly, polyploidy, which occurs when an organism has the wrong number of chromosomes, occurred. Ultimately, these polyploid cells were reproductively dead, resulting in very little cell duplication. Thus, because of these results, microbeam radiotherapy proves to be an alternative radiotherapy treatment for patients with DIPG, because it is more damaging for cancer cells and less damaging for normal tissues. While DIPG is still under research, The current research being conducted by scientists have led to major breakthroughs about the causes and potential cures for the cancer. There's still a lot of work that must be done, but a future cure for DIPG is currently in the works. Thank you for listening.